Thank you, Ann, and good morning. It's my pleasure to be here, and I'm very excited to represent the biopharma industry for the MBDA. Echoing the themes of what you heard from the previous speakers, this is an unprecedented, exciting time in biomarker science. It's enabling new insights into disease biology and helping us to identify new targets, new opportunities to discover new medicines to address patients' unmet medical needs. The biopharma industry, for whom I'm representing today for the MBDA, is very much invested in and committed to a precision medicine paradigm, as you heard before, bringing the right drug to the right patient. However, to do so, we must face many, many challenges, and many of the aspects of this you heard already. I want to draw the parallels between the complexity of biomarker development and assuring the confidence that the biomarker is measuring what it's intended to measure to the same complexity of drug development. And we have to apply the same principles and practice of comprehensive investigation and rigor to the process. Again, from speaking in terms of the end-to-end process. So I'd like to cite one personal example that we had at Bristol Myers Squibb, and it's probably familiar to most of you in the audience. But I think it's an illustrative case study for how to go about instituting the standards that we're speaking of today. In 2008, a number of publications and presentations revealed that approximately half of patients with advanced metastatic colorectal cancer had tumors that contained a mutation called KRAS in their tumors. And the observation was that the patients that harbored that KRAS mutation were not responding to the drug Erbitux, a drug that was already being marketed and available to patients by Bristol Myers Squibb and our partner Lilly. Now, the interesting feature is that the science emerged after the drug was already on the market. And all of these observations, although reproducible and were replicated in many settings and across the globe, they were all done through retrospective analyses of existing clinical data. So how did we have the confidence that the biomarkers that were being used across those studies, as they were reported in 2008, were measuring what they intended to measure with high reliability and reproducibility? So in consultation with the FDA, BMS, its partner Lilly, and a diagnostic partner, QIAGEN, agreed on a program to develop and validate a KRAS test that ultimately received approval in 2012 as a predictive biomarker to help physicians identify those patients who are candidates and would most benefit from the drug Erbitux. It was this type of paradigm, I think, that represents the application of the standards for reliability and for confidence. Going forward, current technology, as you heard, instituting the new biology into clinical trial approaches such as I-SPY2, we're going to continue to identify potential patient selection biomarkers with increasing quantity and rapidity earlier in a drug development process. So what we're excited about from a biopharma industry perspective is that applying the standards that we've spoken of this morning through the MBDA, it provides that platform by which we can all the stakeholders involved, most importantly the patient, but industry, academia, government, payers, can all come together and engage in a very important dialogue to construct a consensus approach to identifying, defining, and implementing those standards to the biomarker development process that ultimately will bring new medicines to patients for the future.